Right. Today we should be talking about the biochemistry of the senses, of the sensory organs, and what kind of signaling pathways or what kind of signaling mechanisms there are in the sensory organs. In lecture, I've divided the senses into physical senses and chemical senses. Which ones are which? Do you understand the division, what the division is based on? Okay, so physical senses detect changes in physical quantities and chemical senses detect changes in chemical composition, basically. So which of the classical senses, or even the other ones that we may touch upon a little bit in the lecture, belong to the physical group and which to the chemical group?
Touch is physical.
Okay, very good. Touch, definitely a physical sense.
Probably hearing as well.
Very good, yeah. Hearing is a physical sense.
Eyesight.
Okay, vision, eyesight. It's physical.
Taste as well.
Taste is what?
Chemical.
Okay, that's a chemical sense.
Smell is chemical as well.
Indeed, yeah. So we had touch, hearing, vision, taste, smell. Do we have smell? Well, yeah, I think we did. Anything else? Balance. What kind of a sense is balance? It's a physical sense, yeah. Okay, so hot and cold are what kind of senses?
Physical.
Indeed, yeah. Okay, changes in temperature. Okay. With pain, we're not going to talk explicitly about pain. Yeah, I'll just leave it at that. We're not going to talk about pain because it does sort of fit into this kind of scheme, but it's not considered a sense generally because it's just a direct irritation of nerve endings usually. So I'm not going to talk about that, but yes, you could argue that it would fit somewhere here. It would be a physical sense generally, not always, but it's kind of in between. All right, any other ways in which we can perceive the environment? I think that's pretty much it. At least those are the ones we're going to talk about. With taste and smell, I'll just say that apart from the typical senses that are considered as taste and smell, we do have cells that basically perceive or that react to changes in nutrient concentration that are elsewhere in the body. So they may not be on the tongue or in the nose cavity, but they can be elsewhere in the body. For example, in the gut, where they actually react to the amount of nutrients, which then causes different kinds of signaling, and it actually influences metabolism, etc., etc. We're not going to talk too much about those, but just be aware that there are actually chemical senses in a way which we normally do not consider typical senses. They are elsewhere in the body, but they also react to changes in the chemical composition of the of the body, or of the blood or whatever. Right. As you will see in the lecture, there are senses about which we know quite a lot, and then there are lots of senses where the biochemistry, the actual functioning of the senses, is still quite poorly understood. So I'll start with the ones that we kind of understand reasonably well and move on to the ones where I'll be just able to say a few things, or maybe just general principles, not the on the details because they're just not known. I will start with the physical senses, and then we're going to finish with the chemical senses. Right. Probably the best understood and the most studied physical sense is vision. The reason for that is, first of all, that it's quite an important sense for us humans, but also, as you will see, it's actually one of the simpler ones. So the mechanism of vision is quite well understood, mainly because it's relatively simple. What is the sensing organ for vision? Okay, so the rods and cones. Rods and cones are actually the sensors. The organ is the eye or the retina. That could be the tissue or something, but correct. So rods and cones are the actual cells that detect changes in light, or detect the absorption of light, really. In the next, I'm going to be talking mostly about rods, but most of the mechanisms are actually the same in the cones. What's the difference between rods and cones? Or what are the differences between rods and cones?
Rods is only for black and white or...
Okay, so rods are monochromatic. Okay, they do not perceive color.
Cones are for more accurate and color vision.
Accurate color vision, okay. So rods are monochromatic, that's right. They don't actually perceive different wavelengths. They only have one wavelength, pretty much. While cones, there are 3 different kinds of cones for 3 different colors, basically. So that's right, that's one difference.
They also have this different morphological, like...
They do look different, yeah. They do have different morphology. There's still one more functional difference. Yeah, they have different positions in the retina, as you will know, but...
They're interconnected to different kinds of people.
They are connected to different kind of processing cells later on in the retina, that's right. There's still one more quite important difference in the parameters of light perception. So one is monochromatic versus color. I mean, to be fair, cones themselves are monochromatic as well, because each type of cone only perceives one wavelength. So the cones themselves don't actually perceive color. But we have 3 different kinds, so we kind of, we synthesize, our brain synthesizes color based on this. So you could say that both cones and rods are monochromatic, only we have a few more types of cones. But there's one more difference in how or what kind of light they perceive. Is there a difference in sensitivity?
Probably.
Probably, yeah. I wouldn't be asking if there wasn't. Yeah, rods are much more sensitive to light. So they are generally used, in humans at least, they're generally used to detect dim light. So rods are very important when it's dim, at night, for night vision. While cones are much less sensitive, so they're used for normal day vision, okay? So the sensitivity is also important. Now, why would the evolution develop cells that are less sensitive? I mean, surely it would be much better if we would be able to see, everything with more sensitivity. Why would we have cones, or why would we have cells that are actually less sensitive to light than rods? What is it?
Faster processing, so that you have a first idea, or what do you...
That's a good question. I don't know if they have faster processing. I doubt it. It's possible. I doubt that that's the reason. What is the point of having cells that are less sensitive when we could have cells that are much more sensitive?
Because probably they receive different frequencies of...
They do, to some extent, but that's not really the reason.
So you have like a day and a night vision?
Okay.
During the day it would be too bright, so...
Exactly. Okay, so the reason for having basically less sensitive cells is that rods, during the day, are overwhelmed with the amount of light. So actually, we can't really use rods because they're just so sensitive that, under normal conditions when there's plenty of light, they basically don't work. They're just overwhelmed with the signal. Okay, so that's why we have cones that are much less sensitive, so that we can actually use them in bright daylight. Okay, so that's the reason for having two different cells with different sensitivities for light. Okay, right? Because very often people think, you know, it's always better to have more sensitivity, but actually, it may not be. Because the difference in light intensity, or the amount of light around us between night and day, is actually many orders of magnitude. Okay, it's massive difference. So we need to have two different sensors for that. Right. So let's now talk a little bit about how rods and cones as well perceive light. You've had retinal structure. You've had lectures on that. Yeah. Okay. So this will be quite familiar to you. This is kind of the shape of a rod. Okay, there's mitochondria there. Okay. Yeah. I'm sure histologists would be able to draw it much better than I. But anyway, you understand the basic structure. What are these things? So this would be the outer segment, and what are these structures there? What are they for?
That's the sensory part.
It is the sensory part.
So it has the rhodopsin with the retinal.
It does have rhodopsin, but what are these structures?
The vitamin A structures.
Sort of, yeah. These are actually parts of membrane, of the outer membrane, which kind of forms these kind of invaginations. And then it forms organelles, membrane or pseudo-organelles. So basically it detaches from the membrane. And you're correct in saying that these membrane-bound vesicles, basically, or pseudo-organelles, contain the actual sensitive protein, the light-sensitive protein, which is rhodopsin, which you're going to be talking about in a minute. Well, why do you think we have structures like this? Why not just have the rhodopsin bound to the outer membrane? That's right, okay? So the point is to create as big a surface area as possible so that we can cram in as many rhodopsin molecules as we can, okay? So these discs are there to basically increase the surface area of the membrane that can carry rhodopsin. Right, to kind of complete the structure, or sort of complete, semi-complete the structure, there are some other cells in the retina which are called, there are many different kinds of cells, but what kind of a cell is this? Bipolar, yeah. But there are other types, as you will know. And then, so this would be one layer, the other layer, and the third layer contains what kind of cells? There are glial cells in there as well. Ganglionic cells, okay? So very simplified structure with rod or cone, bipolar cell, and ganglionic cell, the three different layers. Of course, there are many other kinds of cells, but these are the ones we're interested in. Now, which way in a human or mammalian retina, where is the light coming from? From the top or from the bottom? Okay, yeah. So just to make sure we all know that, there are other organisms that actually have it the other way around, but in mammals, this is the way it works. So the light-sensitive cells are actually in the deepest layer of the retina. Or, well, it's not quite the deepest, but anyway, there's the pigment epithelium here. Right, so this is just the basic structure. Now let's talk about the actual process, the actual biochemistry of light perception. As you correctly said, the light-sensitive protein in rods is called rhodopsin. Rhodopsin is a protein molecule that is very closely related to G protein coupled receptors. Do you all remember what G protein coupled receptors are? Or could you name an example of a G protein coupled receptor? Any?
Specific one?
Yeah. Like a receptor for something.
Glucagon?
Okay, yeah. Yeah, glucagon has G protein coupled receptors. Catecholamines, adrenaline, noradrenaline, all these, serotonin, dopamine, they're all G protein coupled receptors. So rhodopsin looks very, very similar to just your box standard G protein coupled receptor. It has 7 transmembrane domains, all these things. Okay, so it is basically a G protein coupled receptor that is being used to detect not a chemical substance such as glucagon receptor or whatever, but to detect light. In order to be able to do that, it contains a prosthetic group called retinal, which is an aldehyde derived from vitamin A, from retinol. Okay? So this should all be familiar to you. Now, how does it actually work? How does the complex of opsin and retinol, so opsin is the name for the protein without retinol. Okay, so these two form rhodopsin. So how this complex of opsin and retinol can actually perceive light. I'm going to draw the structure of retinol as it is in rhodopsin. You don't obviously have to copy that. You can find it anywhere and you don't need to know it. But I'm just going to draw it so that I can show the actual mechanism. So this is the structure of 11-cis retinol. 11-cis retinol. Why is it called 11-cis?
Because cis is the 11th carbon.
That's right. So this bond has the cis configuration. Okay, all the other ones are trans. This one is a cis. Okay? So this is called 11-cis retinol. This is the connection to opsin. So there is a lysine residue, and the amino group, the epsilon amino group of lysine, binds to the aldehyde of retinol. So it's actually connected to the protein. It's bound to the protein. Okay, so that's how it looks inside rhodopsin. Now, what can you say about 11-cis retinol when you see it chemically? Is there something interesting about it?
It has the group of coupled double bonds.
Okay, we don't really call them coupled.
Conjugated.
Conjugated, very good. So there's a system of conjugated double bonds. All these double bonds are conjugated, which means that basically the electrons are delocalized along the whole molecule. Okay? Now, what is usually, or when we have a long system of conjugated double bonds, what does it often cause, or what does it usually do to the compound? It changes one of its physical properties.
Doesn't it stabilize it?
It does stabilize it. Very good. Very often it makes it colored. Okay? Because many or normal double bonds between carbons would absorb light in the UV region, in the ultraviolet region. But if you have many of them conjugated like this, the energy decreases and these compounds start to absorb light in the visible region. And that's exactly what we want. We want a compound that absorbs visible light, not UV light. Okay? Now I'm not saying that retinol does not absorb any UV light, but what we're interested in is the visible light. Okay? So this is the structure of 11-cis retinol. It's bound in rhodopsin, and thanks to the system of conjugated double bonds, it absorbs light. The maximum absorption for rhodopsin, I think, is around 500 nanometers. So it's kind of a yellowish green light where the sensitivity is the highest. So that's the absorption peak for rhodopsin. Right, so what happens when this structure absorbs a photon? the thing that absorbs photons in chemical structures, in organic structures, are the electrons. Okay, it's the electrons that absorb the energy and something happens to them. So after a photon is absorbed, this whole delocalized system gets to a higher energy. And thanks to this higher energy, this double bond is basically free to flip over to the other isomer. So this double bond suddenly can overcome the activation barrier and can flip from cis to trans. Okay? This is only a probabilistic thing, so it only increases, the absorption of a photon only increases the probability that the flip will occur. So it doesn't have to, but increases the probability. I think the probability in rhodopsin is about 50%. So there's about 1-on-1 chance that after absorbing a photon, it will flip to the other isomer. But it's the flipping to the other isomer that we're actually interested in, that actually causes all the other steps that we're going to look at. So what we want to see is that this 11-cis retinal changes to its isomer, which is, it's probably not going to fit here, but so we're changing the cis configuration to trans configuration. Okay. Etc. Okay. So this cis bond is changed to trans bond. Now, you can already see just from the way I drew it, so this is going to be called all trans retinol, because all the bonds are trans. Now, already from the way I drew it on the board, you can see that the shape of the molecule is quite radically different. Okay? You can basically see that by flipping the bond, the whole molecule kind of stretches out. It becomes longer than it was before. Okay? It was kind of bent before. Now it's straight and long. Okay? This is what, yeah?
I was wondering, is there really any chemical reason why the all-trans is at a higher energy level than the 11-cis?
It's actually not the all-trans that's at a higher energy level. They're both at the same energy levels. But there is the intermediate step, which I'm not showing, which is at the higher energy level. Okay? So basically, this molecule absorbs a photon. It creates a kind of a metastable chemical that can flip either way. Okay? And then it kind of drops down to the base level again. And it either ends up being, again, 11-cis, or it becomes all-trans. Okay? So these two have the same energy levels, or pretty much the same energy levels. Right, so as I said, the absorption and the isomerization of retinol in rhodopsin changes not only the structure of the actual molecule, but also the structure of the protein, because suddenly it doesn't fit into the pocket where it's supposed to be bound. So two things happen. One is that the conformation of the whole protein changes, the configuration changes. Okay? The protein changes shape. The other thing that happens is that the all-trans retinal is released from the protein. Okay? So this bond is hydrolyzed and is released from the protein. So we end up with all-trans retinal and opsin that is still bound in one of these vesicles. Now, the all-trans retinal that's been released from the protein is then recycled by special enzymes, isomerases, that are both present in the rod and also elsewhere in the eye, in the retina. Let's not go into details. So it's basically recycled back to 11-cis retinal, it's bound back to rhodopsin, and we can have another cycle of activation. Okay, so this is what happens to retinol. But obviously, we're much more interested in what kind of signals we get from this mechanism. As I said, the isomerization of 11-cis retinal to all-trans retinal changes the conformation of the protein. And there, we basically get back to the signaling cascade of G-protein coupled receptors. So what happens when a G-protein coupled receptor binds its ligand? What kind of cascade is started? Because the mechanism is exactly the same here. There is a
kinase cascade?
Not with G-protein coupled receptors, or at least not from the very beginning. Does anyone remember how it works? Okay, so the activation of a G-protein coupled receptors changes its conformation. This G-protein coupled receptor is, well, as the name suggests, is bound to a G-protein. The G-protein exchanges GTP for GDP, so it binds a molecule of GTP. GTP? What is GTP? What does it stand for? Okay, guanosine triphosphate. Okay, guanosine triphosphate. So a molecule of guanosine triphosphate goes in, a molecule of guanosine diphosphate goes out, and this activates the G protein. The G protein then dissociates into its alpha subunit and a beta gamma dimer, and both the alpha subunit and the beta gamma then basically start other kind of signaling cascades. Okay, so that's the general working of a G protein coupled receptor. What we see in rhodopsin is exactly the same thing. So the isomerization of retinal activates rhodopsin, and rhodopsin is coupled with a G protein that's called G alpha, so that's the alpha subunit, G alpha T. And the T stands for transducin. Transducin because it transduces the signal. Okay, so transducin. Now, does anyone remember what possible signaling cascades can be started by G proteins?
PKA.
Well, not directly. So the G alpha subunit then goes on and activates or deactivates a protein. Very often, for example, it is adenylate cyclase. Okay, it's an enzyme that changes ATP to cyclic AMP. Okay, there are some other ones as well. Here, however, it's not a cyclase, it's not a protein kinase, but actually the enzyme that is activated by transducin is a phosphodiesterase. And what does a phosphodiesterase? Do you want me to spell out the name? Phosphodiesterase? What does phosphodiesterase do? Okay, so it cleaves phosphodiesters. Okay, do you know any phosphodiester? Not really. It's actually the phosphodiester means that there's a phosphate group that is bound to that basically has two ester bonds. So it's phosphodiester. Is there any compound that looks like that? There are actually quite a few, but well, for example, cyclic nucleotides. For example, cyclic AMP or cyclic GMP have a phosphodiester bond. Because the phosphate, the cyclic thing about it is that it's bound to two hydroxy groups. That's what makes it cyclic. Okay? But it also makes it a phosphodiester. Okay? So in this case, this phosphodiesterase is specific for cyclic GMP, and it hydrolyzes it to GMP. Okay? So rhodopsin's activated, G-alpha T transducin is activated. This activates a phosphodiesterase, actually a cGMP specific phosphodiesterase, that hydrolyzes cGMP to GMP. Now, how do we translate this hydrolysis of cGMP to a signal? Well, in rods and in cones as well, there are, in the membrane, there are ion channels, actually sodium channels, that are sensitive to the concentration of cGMP. So cGMP opens these channels. Okay? So we have cGMP sensitive sodium channels in the membrane. Therefore, if we decrease the concentration of cGMP, okay, I'll just wait for you to write down what you want to write down so you can. Okay, so if we decrease the concentration of cGMP in the cell, what's going to happen to these channels? They'll close. Okay? What will happen to the membrane potential?
get hyperpolarized?
Very good. It will go up. The cell will hyperpolarize. Okay? So, to put it all together, a photon is absorbed by 11 cis retinal in rhodopsin. That isomerizes to all trans retinal, is released from the opsin part. This change in conformation activates a G protein, which is called transducin. The activated G protein, or the alpha subunit, activates an enzyme called phosphodiesterase, which hydrolyzes cGMP to GMP. This hydrolysis, this decrease in the level of cGMP, closes cGMP-sensitive sodium channels and hyperpolarizes the cell. Okay? So here the mechanism is really counterintuitive, or it's unusual in a way that when we want to activate a cell, usually we depolarize it. That's the usual way it happens. Okay? Here, it's the other way around. So when the rod or the cone are activated, they hyperpolarize. Okay? So a very different mechanism from the usual way of activating cells. So does all of this, the whole cascade, make sense? Yep.
And how does, what does the hyperpolarization do? How does it, register that and send the signal?
We'll get right to that. Okay? The other maybe important thing to mention is that, of course, just closing sodium channels on their own would not hyperpolarize the cell. We need to have something to get the potential up, because otherwise it would just stay as it was. What actually happens is that there are pumps elsewhere in the cell that are continually pumping sodium out. Okay? So when we normally, in the dark, when there's no light, there's sort of a current flowing through the cell. Okay? Here the sodium goes in, and here it's pumped out. And that's what keeps the potential quite low. When we close these channels by decreasing the cGMP concentration, the membrane potential goes up because obviously sodium is being pumped out all the time. Okay? So once again, the activation of rhodopsin causes a hyperpolarization of the cell by increasing the hydrolysis of cGMP. Now, a very good question, how does the body, how does the brain register this kind of signal? Because normally the signal is depolarization, and then we know what happens because we release a neurotransmitter, another neuron is depolarized, etc., etc. How does it work with hyperpolarization? Well, when there's no light, so in the dark, the rod continuously releases a neurotransmitter to the bipolar cell. Does anyone want to guess what kind of neurotransmitter it is? It's glutamate. Very good. So glutamate, the most common excitatory neurotransmitter, is being released all the time. Now, when the membrane hyperpolarizes, what will happen to the release of glutamate?
Increase.
When it hyperpolarizes, what is the usual, how does it usually work?
Well, it would depolarize once and then kind of just stay there, and go down again.
What do you mean?
Well, because if we increase the membrane potential,
Yep.
then the cell would depolarize at least once, logically.
Why would it depolarize?
Well, because we would probably reach the threshold of depolarization when we increase the polarity of the cell.
Not really, we're actually moving away from it. As the membrane potential increases, we're moving away from the threshold for depolarization.
But wouldn't we at some point have crossed that threshold?
Oh, I see what you're saying. Not really, because basically the cell, I think what you're talking about is action potential. Okay? We're not really talking about action potentials here. Okay? We're just talking about the membrane polarization. That's all we were really interested in. So we start in the dark. Okay? So this would be the dark polarity. We start with something like this, and then when light goes on, the membrane potential just goes up. Rather, becomes more negative. Okay, that may be a bit confusing. Okay, it becomes more negative. It actually goes down. But yeah. So what happens when it's activated? The membrane is hyperpolarized, and the release of glutamate goes down. It stops. Okay. Now, how do the bipolar cells then transmit this signal further on? Well, they do it by basically there being two different kinds of bipolar cells. There are cells that are called on bipolar cells, and then there are bipolar cells that are called off bipolar cells. The on bipolar cells become activated when the rod becomes activated. Therefore, they're called on. The off bipolar cells become deactivated when the rod becomes activated. Okay? On, off. So there are two different kinds of them. How do you think on molecular level, how is it going to work? How is it going to work that we have two types of cells that basically react differently to the same kind of signal? What would be the possible way of different receptors? Very good. Okay? So the on cells, well, let's start with the off cells. They're easier. The off cells contain the typical ionotropic glutamate receptors, which are, do you know any ionotropic glutamate receptors? Do you know any glutamate receptors?
Nicotine.
Those are acetylcholine receptors.
Oh.
Okay. The three groups of ionotropic glutamate receptors are NMDA, AMPA, and kinase. Okay? The ones here are mostly AMPA and kinase receptors in the off cells. So these are ionotropic receptors. What does an ionotropic receptor, what does it mean when I say ionotropic receptor? Yeah, it's an ion channel. It's a ligand-gated ion channel that opens when the ligand binds. Okay, so going back to our whole thing. Okay, the activation of a rod causes that the release of glutamate is stopped. Now, when there is glutamate, the AMPA receptors will be activated, and the bipolar cell will be depolarized. Yeah. Does it make sense? I'm feeling like I'm losing you a little bit. Okay? When the release of glutamate stops, the bipolar cell that has AMPA kinase receptors will become hyperpolarized. Normally it's depolarized because these are excitatory receptors. Okay? When we stop it, it becomes hyperpolarized. Therefore, it's an off cell. How is it going to work with the on cell? What kind of receptors are we going to, what type of receptors are we going to have there? They are metabotropic, very good, but they have to be inhibitory. Okay? Do you understand that? So these are receptors that activate the cell when glutamate is present. Here we're going to have receptors that deactivate the cell, that inhibit the cell when glutamate is present. Okay, they're called metabotropic glutamate receptors. The abbreviation is mGlur. Okay, a pretty simple abbreviation. I'm not going to go into details what types they are. It's not really important. But they are metabotropic glutamate receptors. And these are inhibitory receptors. So, once again, in the dark, glutamate is being produced. The off cells are going to be activated because they have these receptors. And the on cells are going to be inactivated because they have inhibitory receptors. When there is light present, or when the light strikes the rod, it's going to switch around. So suddenly, glutamate is not being released because the membrane is hyperpolarized. Therefore, the off cells are going to shut off. They will stop being activated because there's no more glutamate present. While the on cells, on the other hand, will actually depolarize because the inhibitory effect of glutamate has been lifted. Okay? Well, and this signal from the off cells and the on cells is then passed on to the ganglionic cells and then passed on to the brain. The reason why we have two different kinds of bipolar cells, some are on and some are off, is that quite a bit of the signal processing in the retina actually occurs still in the retina, not in the brain. So basically the retina itself is kind of a pre-processor for the signal. And having two different kinds of cells on and off actually is part of a circuit that processes the signal and then takes it over to the brain. Okay, I'm not going to go into details why, what the circuit actually is, but that's the reason why we have two different kind of bipolar cells. Okay, for the signal processing that already happens there. Because otherwise we would just be fine with on cells, because that's after all what we're interested in. Okay, but the off cells actually play important roles as well. Right. That's mostly it for vision. Do you have any questions? No. Okay. We've been talking about rhodopsin, which is in rods. Cones have also a light-sensitive pigment, or three different types of light-sensitive pigments, which are called photopsins. Or sometimes you can find iodopsins. Okay, so photopsins or iodopsins are the pigments in cones. This iodo doesn't mean that they contain iodine. It's actually based on the color, kind of purplish color, which in Greek is, iodos. So iodopsins or photopsins are the pigments in the cones. And there are three different types for three different wavelengths and three different colors. In addition to rods and cones, there is actually, as it became clear in the past maybe 5 years, there's one other type of light-sensitive cells in the retina. They are a subtype of ganglionic cells that contain a third type of photosensitive pigment that is called melanopsin. So this is kind of a new finding, that some of the ganglionic cells themselves can actually perceive light. They are very sensitive, because melanos meaning dark, so they're very sensitive. They actually perceive light in the dark. And their purpose there is not to create basically pictures for our brain, but the purpose of these cells seems to be, first of all, synchronizing rhythms. Okay, because that light actually synchronizes our circadian rhythms. So that's one purpose. The other purpose is mediating some of the reflexes, like pupillary reflex, okay, in reaction to light. This has been found because some of the blind people that have complete degeneration of rods and cones, they still have normal day rhythms. They still have normal pupillary reflex. So people were thinking, how is it possible? And then they found out that some of the ganglionic cells themselves actually have a different kind of light-sensitive pigment, and that's how it works. Right. If there are no more questions, let's have a 5-minute break, and then we're going to go through some of the other senses. Okay? So this was a little bit about vision, the best understood of the physical senses. There was quite a lot of it, but I'll promise that actually with the other senses it's going to be much quicker, because we know much, much less about those than vision. So the other group of senses, and it's going to be a group of senses, is going to be mechanoreceptors, receptors for mechanical forces. Okay? Now once again, what kind of senses would fit under the umbrella of mechanoreceptors? Anything that detects any kind of force or displacement. What senses would fit in there?
Balance.
Yeah, balance, definitely. Touch. Hearing, absolutely. Okay, so basically most of the other physical senses that we talked about would fit under mechanoreceptors. Proprioception, all these senses are in fact mechanoreceptors. Now, how do mechanoreceptors work? Again, the details are still unknown for most of them, but generally the idea is that that mechanoreceptors are actually ion channels that are in some ways deformed when a force acts on them, and the deformation opens the channel. the simplest model for such a channel are osmoreceptors in bacteria. What is an osmoreceptor?
Osmo? Osmo?
No, good thinking, yeah, etymologically, but here it's a detector of osmotic pressure. Okay, but yeah, I understand how you think that. So these are detectors for osmotic pressure. Because bacteria need to be able to change their basically internal composition based on the outside osmotic pressure. Okay, because bacteria, unlike us, where we can actually keep our osmotic pressure of our internal fluids in a very small range, bacteria can't really do that. They're dropped into water or salt water, and etc. So they need to be able to measure the osmotic pressure and react to it. So imagine this is an ion channel that is basically just connected to the membrane. So this is the membrane, okay? And it's closed as it is. But what happens if we put a bacterium in an environment that has lower osmotic pressure, that is hypotonic? What will happen to the cell?
It will expand.
Yeah, it will swell, okay? Because water will go in, and the cell will swell. So what happens to the osmoreceptor? Well, as the membrane is swelling, it basically pulls onto the ion channel. And as it pulls, it opens it. Okay? So we can get ions or other chemicals flowing in and out, and the cell can react to those movements. Okay? It's quite likely that we have these kind of or similar kind of osmoreceptors in our brain, where the brain, the hypothalamus, needs to detect the osmotic pressure of the blood so that it can then regulate the osmotic pressure. What hormones regulate the osmotic pressure of the blood or the... Okay, vasopressin, antidiuretic hormone, if we're talking about the brain. Yeah, aldosterone, of course, as well, but that's a bit more complicated. Okay, so the secretion of antidiuretic hormone is probably regulated by something like this in the brain. But this typically is present in the bacteria. Actually, in the bacteria, what happens is when these kind of channels open, is that the small molecules that are present inside the bacterium flow out. Glucose, amino acids, ions, they get out, which basically balances out the difference in the osmotic pressures between the inside and the outside. So they basically dilute their inside so that they don't swell anymore, because then they would obviously rupture. Okay? So this is an interesting and relatively simple model of a mechanoreceptor. So this kind of mechanoreceptor just measures the tension on the membrane. Okay? When it becomes too big, it opens and does various things. Now, in the other senses, such as touch and balance and hearing, we also have ion channels that react to some kind of forces. But here, what we need is it's basically an ion channel that is usually attached to either the cytoskeleton or the extracellular matrix. Okay? The attachment is called tether, which you may have heard before. The name, that's just a general name for something joining something. It's not a specific name for this one. So either it can be tethered to the extracellular matrix or to the cytoskeleton inside, or both, indeed. Now, what happens when we push onto the membrane? Well, as it is tethered, as the ion channel is tethered to the extracellular matrix, as we push on it, the tether will actually pull onto the ion channel, and that will open it. Okay? A similar mechanism probably works in the hair cells in the inner ear, where, as you will recall, the hair cells have stereocilia like that on their surface. Yeah, stereocilia. Okay? And it seems likely that there are ion channels similar to these in the stereocilia, and these channels are tethered to each other. Okay? Or maybe to some other parts of the stereocilium. It's not clear whether they actually, the two channels are tethered together or not, but that doesn't really matter. Now, when we hear a sound, or when we change position in the balance organ, the stereocilia become deflected one way or the other. Again, what happens when they become deflected is that the tether basically pulls on this channel and opens it. Yeah, does it make sense how it works? Yeah, as you deflect it, the tether starts pulling on one of the channels. What is quite interesting in this mechanism is that if the stereocilia move the other way, the signal is going to be different. Because in this case, it's this channel that's open. This one isn't. If we move the other way, this one becomes open and this one closes. So actually the cells can detect which way the deflection goes. Okay? Right. The precise identity of these kind of channels is still unclear, but they seem to belong to two families. One family is called ENaC, which stands for epithelial sodium channels. And the other family is called TRP, or transient receptor potential. Sorry, transient receptor potential. That's not super important, but those are the families in which these kind of channels probably belong. The specific identities are unknown. Okay, so that's very briefly about mechanoreceptors. Okay, there are many of them, they have different mechanisms. The precise identities and precise mechanisms are still unknown.
Are they in free nerve endings as well?
Are they?
In free nerve endings?
They are definitely in the touch sensors. Whether they are in free nerve endings, I don't think so. I think the perception of pain, as I said, is actually not caused by these kind of specific mechanisms. It's kind of a general damage very often. And very often pain is elicited by some chemical substances, protons and potassium and stuff like that. But we're not going to go into that. Okay? Right. Do you have any questions about mechanoreceptors? If not, let's move on to the last group of physical senses, and that's the detection of heat and cold. Okay? The detection of heat and cold is also mediated by ion channels. But these ion channels do not react to physical forces or to chemical substances. Well, they do, as you will hear in a minute. But the primary way of activating these receptors is by changing the temperature. Now, could anyone guess or think about a potential mechanism? How would you design an ion channel that would react to temperature?
Molecules that move faster when it gets warm.
Mm-hmm. Mm-hmm. Well, in a way, that's basically how it is. So as temperature goes up, the movement of molecules increases, okay, as they have more energy. Now, in a protein molecule, as the temperature goes up, the structure of the protein molecule becomes less stable. Okay? Because the various hydrogen bonds and other kinds of bonds basically become easier to be broken. So, as temperature increases, proteins in general become more disordered. Okay? Which is well known because if you boil an egg, that's a denaturation of the protein, which is just caused by that the normal structure is destroyed by the heat, because they become so unstable that they then clump together and form basically a boiled egg. So the same thing happens here. In these receptors, there are domains, they're part of the protein, that are actually relatively disordered. As the temperature goes up, they become more and more disordered. And the bigger the disorder, the bigger the probability of the channel opening. Now I'm talking about the heat receptors. Okay? So the heat receptors are ion channels that have disordered domains, or relatively disordered domains, that when the temperature increases, they become even more disordered, and this opens the channel. Okay? Now, a similar but the opposite thing basically happens in the cold receptors. So they also have disordered domains, but as the temperature goes down, they become more ordered, and that opens the channel. Okay? So the sensing of temperature is basically done by the actual structure of the protein and by the ordering of the protein. As the temperature goes up, things become more disordered. As the temperature goes down, they become more ordered. Here, the identity of the ion channels is actually most likely known. The heat receptor is called TRPV, and interestingly, it's a member of the TRP family that we talked about in mechanoreceptors. So these two kinds of receptors are actually quite closely related. Now this V stands for vanilloid, which you don't have to remember, but vanilloid. And it's called vanilloid because it can be, in addition to heat or increased temperature, it can also be activated by chemical substances from the group of vanilloids. And I'm sure you know at least one vanilloid that activates the heat receptor. Which chemical activates, or which chemicals activate the heat receptor? Are there any chemicals that perhaps cause the sensation of heat?
Alcohol?
Alcohol, not really. It does it indirectly by vasodilation. So that's not really. Nothing that causes the sensation of heat. What are chili peppers? Okay, so the vanilloid that causes the opening of TRPV channels is called capsaicin. Capsaicin, that's the thing that makes chili peppers burn or feel the heat. So TRPV normally is opened by increased temperature, but it can also be activated by capsaicin, which is a kind of vanilloid, which you don't have to really know.
Can I ask something?
Yep.
So basically, it's taste, like the spicy thing isn't taste.
No, it's actually feeling of heat. Okay. We'll talk about taste in a minute, and yeah, the number of different kinds of taste is actually quite limited.
Can I ask? People who eat a lot of spicy food and they're used to it, is it because they damage these receptors, or they become?
It's possible that they've damaged the nerves that contain the receptors, because actually capsaicin has been shown to be neurotoxic in certain quantities and in certain parts of the nervous system. So yes, it could be that they damage the nerves. But also, I think genetically, some people are less sensitive to the effects of capsaicin. So it could be that to start with, they actually didn't feel the pain that much. So yeah, but there's definitely some kind of adaptation. Yeah. Right. The cold sensor has been identified as another member of the TRP family. It's called TRPM8. That's the cold sensor. And that also can be activated by actually several different chemicals. Do you know any chemicals that cause the feeling of cold?
Menthol?
That's right, yeah. So menthol opens the TRPM8 channel.
May I ask?
Yeah.
What would be the function, maybe, like an explanation why those chemicals also make the perception of heat and cold?
That's a good question. I think the explanation is that the plants that produce them basically produce them so that the animals don't eat them. Now, with capsaicin, that's pretty clear. If you have chili peppers, no animal will just voluntarily, apart from humans, no other animal would voluntarily eat them because the sensation is unpleasant in general. With menthol, I think it's probably the same reason, even though with menthol it could be just coincidence. It could be that the plant produces it for completely different reasons, but it just happens to activate this receptor. So I think with capsaicin, it's actually kind of directed evolution. So it makes sense. With menthol, it either could be the same thing or it could be just coincidence. Right, let's move on, unless you have other questions. Let's move on now to the chemical senses. The first chemical sense we're going to talk about is smell, the sense of smell. What is the organ of smell? Is it the nose, really? The whole of it? Cribriform plate is just part of the skull, really.
It's the nerve flow. It's in the superior mucosa.
Okay. It's called the olfactory epithelium. So it's part of the nasal cavity epithelium, but only a small part of it. The olfactory epithelium, that's the actual organ of smell. Okay, so it's not the whole nose. It's just a relatively small part of the epithelium at the ceiling of the nasal cavity. But you're right, there's the cribriform plate, and some of the nerves go directly into the olfactory bulbs, etc., etc. I'm not going to go into details about the pathway. So the actual sensors for smells are olfactory neurons, which express olfactory receptors, or ORs. These are G protein coupled receptors that react to different chemicals in the air. Okay? So ORs are the actual receptors, the sensors are olfactory neurons. Now, different organisms or different mammals have different numbers of genes for olfactory receptors. Okay? The humans have approximately 400 functional genes for olfactory receptors. Okay, so we have approximately 400. We actually have approximately 1,000, but at least 600 of them are not functional. Okay, so we do have other genes, but they don't seem to be working in most people. The fascinating thing is that the differences between the number of genes for olfactory receptors not only differs between different species, like dogs have approximately 1,000 functional, rats have quite a lot of functional genes, but they also differ between individuals. So different people have different numbers of functional olfactory receptors. And that's caused by just different mutation rates. So some of the non-functional receptors, non-functional genes, can become functional because of a mutation, and some of the functional ones can become non-functional because of a mutation. So actually, among people, there are significant differences in the number and the type of olfactory receptors that we actually express in our cells, in our olfactory neurons, which is quite interesting. So that at least partly explains why some people smell certain smells are more sensitive to certain smells. Some people are less sensitive. Some people smell things slightly differently, etc. So there's quite a big variation between species and between individuals.
So this could even change within a lifespan of a person, right?
Possibly. I'm not sure about that because the actual. I'll talk a little bit about how the whole olfactory system is set up, and the setting up is actually dependent on what kind of receptors are expressed. So I'm not sure you can actually change some of the expressions during your life. I'm not sure about that. But definitely in prenatal or in the formation of the zygote, there definitely are differences. So I'm not sure about that. Possibly, but I don't know. Right. So we have approximately 400 genes, and interestingly, each olfactory neuron only expresses one receptor. Okay? In fact, it only expresses one allele of one receptor. Okay, alleles, you remember what they are? Okay, the different copies that we get from the mother and the father. Each olfactory neuron only expresses one allele of one olfactory receptor. Okay, how it's possible, how it's actually done by the cell is still unknown, because this allelic exclusion, that's the process, is still very poorly understood. How is it possible? How does the cell just pick one allele to express? It's unclear. So each receptor expresses only one type of receptor, one allele. The other fascinating thing about the sense of smell is that you have the olfactory neurons okay, in the epithelium, then they go through the cribriform plate to the olfactory bulb. Well, the interesting thing is that all the olfactory neurons that express the same olfactory receptor join together in one place in the olfactory bulb. Well, in fact, actually in one of about 3 places, but they seem to find themselves, even though the receptors can be actually very far away in the epithelium, the ones that have the same receptor then join together in 1 or 2 places in the olfactory bulb. Again, the mechanism by which these different receptors, or this identical neurons, or the neurons with the same gene, even though they are different at different places in the epithelium, how they actually find their way to the common joining place is unclear. Nobody knows that. Okay, but it looks like, and here I go back to that, it looks like the guiding signal is related to what kind of receptor the neuron expresses. So if a neuron expresses OR123 or whatever, then the receptor will kind of guide it to the right place to the olfactory bulb. And it's probable that the guiding pathways were set up during the prenatal development. So that's why I said that if there was a mutation just in the olfactory neuron, it's possible that it just wouldn't find its way to anywhere, because there are no pathways set up for this kind of. So I don't know if it's possible to change it or not. But anyway, so the 2 very interesting things. Each olfactory neuron only expresses one allele of one receptor. And the other thing is that even though the actual neurons can be at different parts of the epithelium, they find their way, their axons find their way to basically the same place or same places in the olfactory bulb. Very interesting. Now, the olfactory receptors are actually fairly promiscuous, which means that one olfactory neuron can be activated by many different substances. Okay, so it's not just one-to-one like you have in serotonin receptor. Serotonin receptor is only activated by serotonin plus some drugs. But here, olfactory receptors can be activated by many different compounds, and even compounds that are chemically quite unrelated. So they are promiscuous receptors. They react to various things. So one compound can activate several different olfactory receptors, but also one olfactory receptor can be activated by many different compounds. So the actual processing of the olfactory signal, of the smell, is a very, very complicated process, because what happens is that, so the smell comes into the nasal cavity, okay? It activates various neurons along the way, okay? And the brain then basically takes all the signals from the individual neurons that have been activated and kind of combines them into one perception that we feel like a specific smell. Okay? So lots of processing, lots of very clever processing must go on in order for us to feel one type of smell. Because several different kinds of neurons will be activated by one compound. Very often, if you smell, a rose or an orange, those are actually those are actually mixtures of huge numbers of various substances. So the smell of an orange is not a smell of one compound; it's a smell of a mixture of compounds. And the brain and the olfactory epithelium must kind of combine all these signals for us to realize that it's actually that we're smelling an orange. So very interesting, still fairly poorly understood. The olfactory receptors are, as I said, a G-protein coupled receptor, or they're G-protein coupled receptors. And the signaling pathways, they activate adenylate cyclase. Okay, so they increase CAMP, and this opens sodium channels which depolarize the membrane and cause an effect. Quite interestingly, they also open chloride channels. Now, I know you're already exhausted, but what usually happens when you open, what happens with the membrane potential when you open a chloride channel? They usually hyperpolarize, because there's usually more chloride on the outside than on the inside. And chloride flows into the cell, which hyperpolarizes it, because we're moving negative ions into the cell. Here, however, the opening of chloride channels actually depolarizes the cell. And the reason here is that in the olfactory neurons, there is a higher concentration of chloride inside than outside. So, I'm just telling you because the usual answer, if somebody asks you, well, what happens when you open chloride channels? The usual answer is, well, the cell hyperpolarizes. Not here, okay? Here when we open chloride channels, the cell depolarizes because the chloride flows out of the cell. Right. Yeah, the name of the G protein is G-alpha, olf for olfactory. Do you have any questions about the sense of smell? It's a fascinating sense, and there are so many things that we still don't understand. Right, let's move on to the last one, and that's taste. Taste, obviously, is another chemical sense. How many different kinds of tastes do we have?
5.
Okay, what are they?
Bitter, sweet.
Bitter, sweet, sour, salty, umami. Okay, so those are the 5 basic tastes. Okay, you've all heard of umami taste? No? Okay, we can talk about that. So these are the 5 basic tastes. However, now it seems like there might be another one, which is tasting fatty acids, tasting fat. There's some evidence that it could be another basic taste. Okay, now we said we only have 5 or maybe 6 basic tastes. So how is it possible that we can taste so many different kinds of tastes in a way? When you eat different kinds of foods, it's not just that we say, oh, this was salty, but we can actually, oh, this tastes like strawberries and this tastes like whatever. Here, the majority of the signal actually does not come from the taste sensors, but from the smell sensors, okay? So what we call generally a taste, or something tastes like something, it's not so much taste, it is a little bit, but it's also smell, okay? So a big part of it is actually smell. Anyway, so we have 5 basic tastes, and for these 5 basic tastes, we have, obviously, receptors. The first 2 receptors I want to talk about are receptors for sour and salty. These 2 receptors, and that's something you might be able to come up with, are actually ion channels. Because the substances that cause the perception of salty and sour are ions. Okay? Make sense? Yeah? So salty is elicited by what? What kind of substance? Well, yeah, but what part of salt causes? Sodium ions, okay? Sodium ions are the prototypical substance that causes salty perception, salty taste. It's not the only one, actually. Potassium, some of the other ions actually cause similar sensations, but it's mostly sodium ions. What about sour? What is the substance that causes sour? Protons. Actually, when you read the literature about the perception of sour taste, there are people who say that it's actually not so much, well, that it is the protons, but it's not the protons on the outside, but it's the protons on the inside of the cell. Okay? I'm just going to leave it at that. I'm not going to go into details, but it looks like it's a bit more complicated than we think. Anyway, we have 2 different kinds of ions, and we want to detect their concentration somewhere. So we use ion channels for that. Now, interestingly, Neither of these ion channels has been identified certainly, with a certainty, in humans, or actually in mammals. So we're not sure which ion channels actually mediate these two tastes. There is some evidence that the sour taste is mediated by a PKD channel, which is related to a disease called polycystic kidneys. You may have actually heard about those in embryology, in the development of kidneys, polycystic kidneys, when the things don't join quite well. So one of the proteins that is involved in this disease might be the ion channel that allows us to taste sour taste. But you don't really need to know these identities. The salty channel is probably another member of the ENaC family, which we saw before as mechanoreceptors, so epithelial sodium channels. But again, specific identity is not clear. So that leaves us with three tastes, with sweet, bitter, and umami. These tastes are detected by relatively well understood G protein coupled receptors. Right. Sweet. What kind of substances elicit sweet taste? Okay, glucose, fructose, sucrose, so saccharides. Are there any other things that? Okay, still pretty much saccharides, but yeah, sugar alcohols are also sweet.
The artificial sweeteners?
Yeah, there are plenty of artificial sweeteners like aspartame. Do you know what aspartame is chemically? It's actually a dipeptide. It's a compound made out of 2 amino acids. So completely unrelated to saccharides. And actually there are lots and lots of different substances that cause, just so that doesn't confuse you. GPCR? Do you know what it means? G protein coupled receptors. Okay, so this is just all 3 are G protein coupled receptors. There are many other substances that cause sweet taste. For example, some inorganic substances as well, like beryllium salts or lead salts actually taste sweet. So, as we said with smell receptors that they are promiscuous, these taste receptors are actually fairly promiscuous as well. There are a relatively large group of compounds that cause these kind of tastes. Okay, so they're G protein coupled receptors and they're fairly promiscuous. The specific types of receptors are, there are 2 types of taste receptors. T1R, so taste 1 receptor, taste 2 receptors. Okay, T1Rs and T2Rs. The T2Rs are receptors for bitter taste. There are about 35 or so many of them, and they detect different kinds of bitter tasting compounds. Okay, so they are the 2 T2Rs. Now, the sweet taste and the umami taste, I'm going to talk about umami in a minute, are both detected by T1R receptors. But the sweet taste is detected by the subtypes 2 and 3, and the umami taste is detected by subtypes 1 and 3. Okay, you don't have to know that, okay? You don't have to know the subtypes, but it would be good to know T1Rs and T2Rs. Right. We talked a little bit about the types of sweet substances, or the substances that cause sweet taste. With bitter taste, again, the group is huge, and they are relatively unrelated to each other chemically. But what you often find is that chemicals that have amino groups, very often tertiary and quaternary amino groups, taste bitter. So if you see a compound that has a positively charged nitrogen with 4 things bound to it, it's quite likely it's going to taste bitter. What about umami? umami was originally discovered as the taste of glutamate. Okay, so it's the taste of glutamate. In fact, the umami taste can be elicited by most amino acids. Okay? So we have sweet for sweet things, bitter for bitter things, and we have umami for the taste of amino acids. So in fact, when we talk about food, you could say that umami is the taste of meat or meat products. Okay? So amino acids cause the umami taste. Glutamate being one of them, but not the only one. Many amino acids actually do that. Final thing, I said that there are G protein coupled receptors. The G protein is called gustducin, which is kind of a play on transducin, which we saw in the beginning, but because it's gustatory signal, it's called gustducin. And it also increases CAMP. All right, and that is all. Do you have any questions? No? Okay. All right. Well, good luck with your exams then.